Welcome to the Protein Synthesis series. This video focuses on transcription. At the end of this lecture, students should be able to describe transcription, its steps, and how mRNA transcripts are developed. The central dogma of biology describes the way in which cells express genes found within their DNA. DNA is the blueprint for the growth, development, and reproduction of organisms, from conception through death. DNA can be replicated to create additional copies of itself for new cells created during cell division. DNA is also used as a template to transcribe into pre-mRNA inside the nucleus. The pre-mRNA undergoes modifications to become a mature mRNA strand ready to leave the nucleus for the cytoplasm. This transcript then is read by a ribosome that translates it into an amino acid chain called a polypeptide. The polypeptide is the protein which determines the form and function of each cell. The protein is expressed by the organism. Humans potentially hold over 100,000 different proteins, all with specific functions. The genome of an organism is the sum total of all its genetic material. Our DNA is contained within 23 pairs, or 46 total chromosomes. Each chromosome contains genes that code for the proteins necessary for humans to survive, reproduce, and interact with our environment. A gene is a functional unit that is composed of nucleotides in a particular order that leads to the production of a particular protein that offers a specific function in the cell or organism. Genes carry our heritable information. They are the traits we see expressed when we look at others and ourselves. There are nearly 25,000 genes in the human genome. That would equate to over 500 genes on every chromosome in your cells. Not all genes are expressed in a visible trait. However, all genes are expressed at some point in our development. Transcription is the act of making a copy of a segment of DNA, including the gene and control mechanisms, into a readable format on the ribosome. The transcript is created and modified into mRNA. mRNA is a messenger RNA whose job is to carry the DNA message, which is too valuable to leave the nucleus, out of the nucleus into the cytoplasm into a ribosome. Each gene contains multiple regions of DNA, some used for coding of proteins, and other non-coding regions for functional control. Enhancers are a non-coding mechanism used to regulate gene expression. They turn transcription on and off as the proteins are needed by the cell. This regulator is similar to a loan officer. A person has too much debt, or in our case, too much protein. We don't need to make more of it. Let's reject it. We'll turn it off. This person has very little debt, or doesn't have enough protein. We need extra protein, so let's push through and make more protein. The next non-coding mechanism is the promoter sequence, which is called the TATA box, because it's always the sequence TATAAA. It comes upstream or before the coding region for the transcription start site. Think about a club promoter trying to get people and bands and DJs to come to a new club, or a restaurant promoter trying to get you to try a new taco. The transcription promoter is trying to get the RNA polymerase II enzyme to come to start a new transcript of mRNA. Next, we see the coding section, full of introns and exons. The introns are thought of as junk DNA that is excised from the transcript. In other words, we don't know exactly why it exists other than to prevent mutations from coding regions forming. The coding region is the region that forms the protein, so it would be a little more important to continue having those segments without DNA mutation. Conversely, the exons are used for the coding of DNA that is necessary for gene expression. So remember that introns are the junk in between the exons, and exons are expressed. Finally, we come to the termination sequence, which is the stop. You are done point. Some enhancers may be found after the stop codon, which have the potential to inhibit or enhance transcription of the gene as well. Transcription has 3 stages: initiation, elongation, and termination. Initiation is the beginning or the start of transcription, when the transcription factors are in place and the RNA polymerase II binds to the promoter site. Elongation is the actual synthesis of the RNA by the polymerase enzyme. Termination is the end or stopping point of transcription, when everything dissociates. DNA is double-stranded and exhibits complementary and antiparallel nature. With regard to transcription, one strand of DNA is the coding strand. The other is called the template strand. The coding strand shares the code with the mRNA. They are identical, except that thymine in DNA is replaced by uracil in RNA. The template strand is the strand being read by the polymerase, and the mRNA complements are built off the template, with the exception again of thymine being uracil in mRNA. Let's give this a try. If the template reads 3' TAC GGG ATA CAC 5', what would the mRNA strand be transcribed as? Because the strands are antiparallel, the 5' end will begin the mRNA sequence. Then we proceed matching the complements to the template. The DNA strand is the template strand. Keep in mind that the thymines are replaced with uracil in mRNA. So our new strand would read 5' AUG CCC UAU GUG 3'. Initiation begins when the transcription factors bind to the DNA template strand, allowing RNA polymerase II to bind to the promoter or TATA box. The DNA is read from 3' to 5', having the mRNA transcript assembled starting with the 5' end. This means that each new nucleotide is added to the growing 3' end of the transcript. Elongation will continue adding nucleotides to the 3' end of the growing mRNA strand, matching the complementary bases C with G and A with U, until the termination sequence is reached. When polymerase interacts with the termination sequence, the mRNA transcript will be released. The polymerase, DNA, along with the transcription factors and enhancers will all dissociate from the complex. The pre-mRNA or primary RNA strand is also called heterogeneous nuclear RNA, or hnRNA, because it contains both expressed exons and junk introns before modification. The pre-mRNA strand now needs to undergo processing to be ready to leave the nucleus. To begin, a 5' cap is added to the newly synthesized transcript. It's used to prevent degradation of the coding sections, as well as providing recognition by the regulatory pores that allow the transcript to exit from the nucleus to the cytoplasm. Next, the 3' end gets a poly-A tail, which as it sounds is simply 200 A's added to the end of the transcript, again to prevent degradation of the coding region, but also to offer stability or longevity for the transcript in the cytoplasm. It also helps with exiting the nucleus through the selective pores. Finally, the transcript undergoes splicing by the spliceosome, where the introns or intervening regions are removed, leaving only the expressed regions for export. The snRNPs or SNRPs are used to stabilize the transcript while it forms loops to splice out the introns, which covalently bond the exons. The finalized product is the mature RNA molecule, known as messenger RNA. Some genes code for more than one protein, and alternative splicing with a different organization of exons is one mechanism for alternate protein coding. The ribozyme is a specialized mRNA transcript that splices itself at the introns that are used for the coding of transfer RNA or tRNA used in peptide synthesis, or for rRNA or ribosomal RNA, which are subunits necessary for reading the mRNA transcript. When the mature mRNA leaves the nucleus, it is finally ready to find a ribosome to translate its protein message. Students should be able to understand the central dogma of biology. Students should be able to describe the 3 stages of transcription. Students should be able to explain the role of the promoter, enhancer, and termination sequences of a gene. Students should be able to contrast introns and exons. Students should be able to transcribe DNA into mRNA. Students should be able to discuss the importance of RNA modification, such as the 5' cap and the 3' poly A tail, or splicing. Students should understand the difference between RNA, mRNA, hnRNA, pre-RNA, mature RNA, tRNA, and rRNA.